Good afternoon. My name is Wes Jackson. I'm one of the co-founders and now the Chief Science Officer of Valitor. Really appreciate this opportunity to come share our updates. Like the last few companies we heard, Valitor's mission is to extend the durability of retinal medicines, and we're targeting the kind of long-standing clinical goal of once every 6-month treatment to fit into the clinical protocols that we're hearing from the clinicians work best with how they want to manage the patients. We're doing it in a way that's a little bit unique. So we have compositions that are designed to passively interact with the vitreous matrix in order to give us a slow, predictable elimination rate from those ocular tissues. Our compositions are designed to be administered through a 30-gauge needle, standard intravitreal injection volumes, and all of our compositions are transparent and dispersive, so there's no risk of optical interference. And these are not depots, so we're not leaving anything behind. The entire complex with the bound targets eliminate from the eye, following endogenous routes. So a good example of how we're doing that is with our lead product, VLTR-559. It's a long-acting anti-VEGF to treat wet AMD. We're choosing to use anti-VEGF because for most patients it works really well. They achieve a treatment maximum. So there's not much more we can do to treat their visual acuity, but all patients could benefit from longer treatment. And so the way we do that is at the core of our molecule, we use a long chain of hyaluronic acid. Hyaluronic acid is endogenous to the vitreous matrix, it's biocompatible, and that's the component that really is able to intertwine with, interact with the native tissues in the eye and give us that slow, reliable elimination rate. To the hyaluronic acid, we conjugate single-domain anti-VEGF antibodies. We like these antibodies because they're small and they have some inherent stability, which is important when you're planning to put these in the back of an eye and keep them there active for months at a time. We want to make sure that the chemical conjugation between the hyaluronic acid and the antibody is covalent and non-labile, because not only does it infer the PK advantages, but we're actually able to coordinate multivalent interactions between the antibodies and their target, so we can see an increase in potency on a per antibody basis. And I just want to highlight one of the key differences of our approach is the overall shape of the molecules that we're generating. So unlike a traditional biopolymer conjugate that may form a sphere, these molecules based on hyaluronic acid form these long worm-like shapes that enable them to thread through the tissue matrix. And so I like to say they're large in the way we want them to be large because there's a lot of molecular mass that defines the elimination rate out of the back of the eye. But they're small in the way we want them to be small because they're able to penetrate into pore sizes and deeper tissues where you typically think only an antibody or an antibody fragment may be able to penetrate. And so we're really excited. This year at ARVO, we're going to present our non-GLP IND-enabling data package. The presentation is going to be on Sunday afternoon. I encourage everyone to go see it. We're going to be able to show the safety profile we've established in non-human primates at the clinical dose and at a 3x tox dose, showing that it was extremely well tolerated. The potency in vivo and in vitro is equivalent to all the other anti-VEGFs currently on the market. And we're able to differentiate ourselves on the basis of our PK profile. We're seeing retention in the ocular tissues on the order of 3 to 4 times longer than something like Eylea, which is more than enough than we need to get that reliable once every 6-month dosing with a pretty decent safety factor. I do want to show one quick highlight. This is the PK results of a rabbit study showing the concentration of the drug as it's eliminating slow and reliably over time in blue. We're also showing that we've got good tissue penetration into the retina and subretinal tissues as well. We're expecting VLTR-559 to go into the clinic around the middle of next year. We just kicked off a bunch of manufacturing activities in order to keep us on that date. We've also had the opportunity to work with a couple of clinical industry collaborators who have clinically validated assets that need durability in their space. And through these interactions, we've been able to confirm both the potency, safety, and durability profiles of these products are shaping up to be very similar to VLTR-559. We've recently expanded our management team, hiring Greg Kunst as our CEO about 6 months ago. He's been helping us recruit new management team members who have the clinical and business development experience we need to continue growing the company. And that's starting to catalyze into good traction on our next fundraising round, which is designed to generate phase 2 data that is really robust and able to demonstrate the capabilities for durability of our lead program. Thank you. Hope to talk to some of you during the breaks.